Welcome to Global Harmonization Initiative webinar series. I'm Dr. Diana Bogieva, the GHI Working Group Director and your host today. Our webinar is focused on genetic toxicology and genomics and is prepared by the GHI Genetic Toxicology and Genomics Working Group, a group working on improving experimental protocols for assessing human risk to different classes of chemicals in air, soil, water, and food, aiming to harmonize and modify state-of-the-art experimental models and technologies for the most effective and reliable biological assay to improve safety standards globally and to improve human health. This webinar presents examples of some of these models. I'm really pleased to announce our wonderful speakers today. Now we have Dr. Firuz Daroudi. He is the chair of the GHI Genetic Toxicology and Genomics Working Group. Also with us today is Professor Dr. Gareth Jenkins from Swansea University Institute of Life Science in the United Kingdom, and Professor Robert Tureski from the Masonic Cancer Center and Department of Medical Chemistry at University of Minnesota, United States. Please, let's welcome together our speakers and yourself being part of our wonderful audience. Together, we'll discover the many aspects of genetic toxicology and genomics. Before we continue with the talks, I wish to thank ICC for providing this webinar platform to enable our presentations and also important and meaningful conversations. Thanks goes to the great effort of the GHI webinar team, including Dr. Nicholas Stanley, our GHI Communication Director, Dr. Gerhard Schleining, our technical guru, who will also help me with presenting the results from our interactive poll. We all are going to be a part of it in a minute. Also, big thanks goes to our audience for joining us today. Please feel free to ask as many questions as possible. So, you can place them in the question area on your screens. And also, as I mentioned, we'll be running few interactive polls. You'll receive the link to these polls on your screen after the Q&A session. Please provide us with the best and prompt responses possible. Let's start with our first speaker today, Dr. Firuz Daroudi. He is the chair of the GHI Genetic Toxicology and Genomics Working Group, now retired, but Dr. Daroudi was previously at Leiden University and specifically working with the University Medical Center, Department of Radiation Genetics and Chemical Mutagenetics and Toxicogenetics at Leiden University in the Netherlands. He developed 4 systems that can contribute significantly to improve food safety standards and to reduce or eliminate the use of animals in testing of different classes of chemicals and in genetic toxicology programs. Dr. Daroudi will shed light into the development and validation of human hepatoma cells in genetic toxicology for assessing food safety, state of the art. It's my pleasure to welcome Dr. Daroudi, and welcome, and over to you.
Okay, thank you very much, Dr. Bogova. I also would like to start my talk by saying good morning, good afternoon, and good evening to all the colleagues that participated in this webinar. The rationale for this GHI webinar is sort of the introduction to the field of genetic toxicology and genomics, and to show you the potential and also very important issue, the objective and mission that I hope that we can reach together to the platform that we can improve the existing criteria for genetic toxicology upon which we can improve the human health. And the topic that I have chosen is to show you also the potential and existing knowledge of what we have for genetic testing is the application of the human hepatoma cells. And the issue is that to address the food safety as much as feasible in a very accurate way and very much relevant to the human. As it's known that food and dietary component is marked one of the main causes of the cancer. And of course, in different countries, we may have different proportion, but as you can see in this figure that up to 10 to 70% of the type of cancer we get might be directly to the nutrition. So this is a very important issue to address, and upon which also indicate that the existing information has not been proven enough to help us in order to reduce this correlation of the nutrition and the high incidence of the cancer. So the way that we are being exposed to either naturally, occupationally, accidentally, or environmentally, the first line is the sort of the radiation of different types, could be sunlight, could be radiation of the gamma or Z radiation, or could be environmental pollution. And other issues that the lifestyle may have an effect as such that it may lead to cellular damage. This cellular damage will be manifested on DNA protein membrane cellular organization, and consequently it may lead to cancer. So this is the very important issue to consider. The result of this type of exposure, the endpoint might be diminishing the human health. So, and of course, there are for years and years, we do have the special guideline regarding genetic toxicology for testing. And this OECD guideline, when you look upon, you will see that it's very much dependent on animal studies for different parameters that the tests are being approached. And however, what we should consider that there are setbacks for this using either existing in vivo and in vitro that you just highlighted very briefly. The first of all, in vivo assay that being used, the intake level are many times higher than likely taken by human. And what it has been done is extrapolation of the data to determine the safety for human, and they employ the safety factor. Consequently, using the high doses long-term experiment, this is associated with high cost. Furthermore, it has been proven and shown that high percentage of both false positive and false negative emerge from this data, and also the data obtained, they were lacking reproducibility. Coming to the in vitro assay, with metabolically incompetent eukaryotic cells like Chinese hamster ovary cells, human lymphocyte, human fibroblasts, as well as the prokaryotic cells in salmonella assay, in order to look for the effect of indirectly acting chemicals, exogenous enzyme homogenate from blood liver or slime mix has been used that is heavily dependent on cytochrome P450 enzymatic activities in rats. And what is has been again evidence that there are significant difference in enzymatic profile between human and rat. Furthermore, certain mode of action of phase 2 enzyme to detect the protection or detoxification process are not represented adequately in existing in vitro models. And this all may lead to the false negative, and in some instances may lead as well to false positive data. So what we have done in many years back is that we have used the human hepatomo cells, particularly in this case the hep2 cells, and here I have highlighted the uniqueness of these cell lines. First of all, human liver origin, so it's directly relevant to human. The mutagenic effect can be studied in the cells that are also targeted to assess the DNA damage. This obviously simplifies the test, and also it allows to study the mutagenic activity of the short-lived chemical. And what we have done in order to expand the utility of the system is that we have generated microsome fraction for hepatomo cells and use it in other cell types, again, Chinese hamster ovary cells, human lymphocyte, as well as M. salmonella assays. And what's remarkably distinctive for the hep2 cells, that what we find, unlike other in vitro cells, that they could possess, even following the cultivation, they maintain the level of the phase 1 as well as phase 2 enzyme. And in the studies performed by my colleague, they have assessed the induction for the phase 1, the responsible for activation, as well as phase 2 enzyme responsible for detoxification, and they made a comparative analysis between human hepatocyte and human hep2 cells. But here I should add it up. Though for the CYP1A1 and CYP1A2, the initial induction level was distinctly lower in human hep2 compared to human hepatocyte, but the induction level following treatment with benzpyrene was equal, as you can see in this figure. So both for the phase 1 as well as phase 2 induced, the level was similar between human hep2 and human primary hepatocyte. This implicates that this could be the useful model in order to be used for assessing the effect of the directly as well as indirectly acting chemical. Furthermore, focus of attention in other studies primarily to the phase 2 enzyme, and as you can see here, for the predominantly the phase 2 enzyme required for detoxification is present, and the level is equal between HepG2 and human hepatocyte. Unlike in two cases for UGT1A1 and 1A6 transcribed, the value in hepatocyte was significantly higher. So by knowing this type of information, this will also allow us that once we get the negative data to emphasize the possibility of lack of the proper enzyme for activation of the chemical of interest in HepG2. And this actually lead also the other type of the experiment as such, the aim to generate the different types of the human hepatocyte cells in which they look for the possibility of the enzyme that is lacking HepG2 or other type of the enzyme that may not be present in HepG2, both phase 1 and phase 2, would be present in those type of cells and used for genetic toxicology studies. So the way that we have performed the work mainly in Leiden and also with my colleague, we have generated battery of test systems. This is a very important issue. We were not dependent on one test in order to assess the genetic testing program, but we have made a panel. And this panel, what aim was that to look for the DNA damage induction and chromosomal alterations. And these mainly chromosomal alterations are known to be biomarker for cancer. So this is a list of the assays we have developed. Shortly, I show you how they look like. The system exchanges micronuclei in binary cells, and then we apply pan-centromeric probe in order to detect whether micronuclei originate from whole chromosome or part of the chromosome. And these are a phenomena we define it as the anogen or the clusterogen. And then we look for chromosomal alteration, standard DIMSA staining. We have looked for structural numerical aberration using the state-of-art technology, generate a probe for individual chromosome or the whole genome. Then the COMET assay look for DNA single-stranded or double-strand breaks, reactive oxygen species, ROS assay, DNA damage induction, repair kinetics using the RAD51 or gamma-H2X, and cytochrome P50 profile for both phase 1 and phase 2 enzyme, and finally using the gene expression profile analysis by Affymetrix microarray. So as you can see, the platform was really approaching all types of the events that may happen in cells following exposure to genotoxicant. So these are some examples. The scenario that we are getting the micronuclei is either the chromosome, all chromosome loss or the part of the chromosome. And in the right side, you can see by DIMSA staining, it can be easily visualized. What we have done in order to see whether it's origin from the whole chromosome or part of a chromosome is application of the centromeric probe, because the chromosome, each chromosome has one centromere at the region that the both chromatids are get involved. And this illustrated the centromeric probe for the whole genome. We even mapped the whole chromosome by putting the telomeric probe as well. As you can see, we know that also telomeres are involved in the aging process. So then we might be able, by measuring the size of the telomere, this can reflect the shortening of telomere, perhaps by doing the treatment with the genotoxic agent. And there are great advantages in this scenario, but having the centromeric probe, we can classify whether the micronuclei originate from whole chromosome or part of the chromosome. And this is an example of the COMET assay, that what we do in our study, we measure the tail length plus tail intensity, and this gives us the tail moment, could be used for the measure of the analysis of the data. And finally, especially what we need for the testing is that we have developed the automatic system, and this automatic system allows us to generate a large amount of the data. And it was not only meant for one type of the biological assay, it meant for series of the biological assay. Examples are illustrated here. The B section regarding the chromosomal alteration, the C illustrated the micronuclei analysis, detection, as well as the scoring. The D reflects the COMET assay, detecting and measurement, and also the E up to H reflecting the spot counting. And briefly, the presentation I make, we are addressing the genotoxic potential of the human dietary component. We try to detect and discriminate between carcinogen and non-carcinogen. We assess the anti-genotoxic potential of the chemical of choice. We also look for mode of action of the enzyme involved in this process. And finally, we hope that by making the packet, complete packet and doing the series of experiments in this regard, we can try to find the alternative system for testing program. So in order to test the suitability of the HepG2 cells in the testing program, we made a comparative analysis between the existing data in vivo using the animal compared to the data obtained with HepG2. And also we checked the structure-related chemicals like carcinogen 2-RF compared to non-carcinogen 4-RF, benzpyrene compared to pyrene. As you can see that we got a very good correlation between the HepG2 result with what in vitro, what we find in vivo using animal. And however, when we look to the potent carcinogen 2-RF using the rat liver S9 mix, it wants to be negative in CHO cells. Though when we use the S9 mix from HepG2, it came positive using the same cell lines CHO cells. And also the take-home message in this regard is that it seems that the HepG2 cells have the potential to detect and discriminate between carcinogen and non-carcinogen. And very interesting set of data that we observed was that regarding to the two chemicals, HEMPA and hexamethylphosphoramide and saffron, both with the rat liver S9 reported negative, either in Ames test or in CHO cells. But when we use the S9 fraction from HepG2, positive data were obtained. Same was held true when we use the HepG2 cells as a target for DNA damage. So after that, we searched a series of the chemical in the two classes of the human dietary component. One is the heterocyclic aromatic amine, and the second was mycotoxin. And this is the list here for heterocyclic aromatic amine and how we get the heterocyclic amine by baking or barbecuing the protein-rich food. And one of the main issues is that these are known existing data reveal that they are mutagenic in pro and eukaryotic organism, and as well as in the animal, it may cause cancer. Furthermore, a previous study indicated they may have been involved in the etiology of the colon cancer in human, and heterocyclic aromatic amine, as already mentioned, has been tested extensively in the salmonella assay by a group of Professor Subimura and was found to be positive. However, the result obtained in a genotoxicity test in mammalian cell lines are highly divergent. We applied this heterocyclic amine at different doses to HepG2 cells, and what I just summary of data illustrated here. We have checked the medium effective dose, that's a dose that frequency of the chromosomal alteration in this regard to macronuclei increased by factor of 2. And then we make a ranking order, and as you can see here, TRPP2 came to be the most genotoxic among these heterocyclic aromatic amine tested. Furthermore, we checked the mycotoxin. Mycotoxin, again, the reason for choosing this class of chemicals we are being exposed to on a daily basis. So the mycotoxin are present in wheat, maize, and then these are the series of the mycotoxin we have tested, except that aflatoxin B1 that known to be carcinogen in all the tests has been done so far. There are no data available in vitro for other mycotoxin of interest presented here. And the existing data reveal, as I mentioned, that primarily the effects shown in the animal, different types of the cancer they have generated in different types of the animal. And very important to know that they are very stable, this mycotoxin. They are stable during the storage and the myelin process and difficult to destroy them under high temperature. And in human, there is no genotoxic data, but epidemiological studies have indicated they may cause esophageal cancer. Negative data using a salmonella assay, as well as no marked positive data reported using in vitro test system. So as long as we are negative, there's no way we can look for mode of action of this chemical. So we have tested this mycotoxin HepG2, and this type of report was for the first time that we reported genotoxic potential of mycotoxin in an in vitro model. And the interesting is that, as again, you can see we made a ranking order based on the minimum effective dose, induced frequency of macronuclei by a factor of 2, compared to the samples. In control samples, the aflatoxin B1 by far it came as number one in this ranking order. Other interesting issue is that when we check ochratoxin B, that is a structure-related chemical, to ochratoxin A, that is carcinogen, and the ochratoxin B is non-carcinogen, we used macronuclei and comet assay. In both assays, we got negative data. Now the issue that I already approached and mentioned that when we have centromeric probe, it gives us the possibility to look for the origin of macronuclei. So when it's coming from the part of the chromosome, you see no spot in the macronucleus. When it's coming from the whole chromosome, you will see a spot in the macronuclei. Therefore, now we can check two parameters. First, induced frequency of macronuclei, and then the frequency proportion of the centromeric plus and centromeric minus. Why? Because centromeric plus indicates that the excess number of the chromosome, whole chromosome loss, and this is the phenomenon we call it anogenic potential. And when the number of the C minus is high, we call them clustergen. And generally, it is proven that anogens are carcinogen. One example is that we check for the citrulline. This compound can be found in more folds, and it causes urinary tract cancer. So with the citrulline, you just look to the proportion of the macronuclei C plus compared to C minus. First of all, we get the induced frequency of macronuclei at different doses, and the proportion of the C plus is about 80%, and this indicates that the citrulline is anogenic. And this again, now we have the system, not only we can classify carcinogen, non-carcinogen, we can also check and discriminate between anogen and clustergen. And of course, we have tested also a series of the human dietary components known to be anti-carcinogen, and the lists are here. And the tests we have done, we have did the comet assay for the STGE, single-cell electrophoresis, or macronuclei. Different types of heterocircular aromatic amine has been tested at different doses. And when you see the positive, this indicates that we find anti-genotoxic potential. In addition to this interesting data, what we have also observed for some two cases of the two vitamin C, as well as vitamin A, beta carotene, that excess of amount of this by itself is genotoxic. So when we use those are higher than the 20 milligram per ml vitamin C, the frequency of macronuclei increased in HepG2. The same was true at doses higher than 10 for the beta-carotene vitamin A, and this gave a cautionary note that some of the vitamins that you may think is helpful always, we should have a cautionary note on the doses that we are using and the period that we are using this type of the vitamins. And we continue the issue of the anti-genotoxic agent, and one of the points was that we have checked the extract from 3 beers, and the lager beer, stout beer, and the Guinness beer, and this illustrated that, first of all, when you following treatment with TRPV2, why we choose TRPV2? Because it came to be number one genotoxicity potential among the heterocircular aromatic amine tested. And by itself, this extract, when you use a dose of 0, has no effect. But in particular, the case of the lager beer and Guinness beer, we find reduction in the frequency of the induced macronuclei that was not present in the stout beer. So then we made a thorough analysis to see the difference. What is the reason for this of the reduction? First of all, check the effect of the alcohol, and we find the ethanol per se by itself is a genotoxicant and increase the frequency of macronuclei. And then when we make a comparative analysis, we find in the stout beer was different compared to the lager beer and the Guinness beer, because that one did not have glycine betaine. So we have extracted glycine betaine and have performed a similar type of studies, and this implicates that when you look at the blue compared to red one, the frequency of induced macronuclei reduced to almost the control level in HepG2, but other cell line that we have put Hep3B is lack p53, and in that cell line we did not see difference, and that implicates the genomic instability present in Hep3B that just abolish the anti-genotoxic potential of glycine betaine. And what we have done further in this was that to try to characterize, to see the influence of this, what is the mode of action of this enzyme involved. And as you can see in this case, it was very clear when we have TFR2 alone, this, especially the phase 2 enzyme, UGT, decreased significantly. But when we have the glycine betaine, the frequency just came back to the control level. So it's an interesting set of experiments that we can look to enzymatic profile induced and involved in the process of the genotoxicity for different classes of the chemical that we are using. And finally, I would like to approach the recent study we performed, the clinical application of the HepG2. This is rather unique, and that actually paved the way toward making the personalized medicine. Generally, all the drugs being applied for cancer patients are being tested in animals, and the doses are being taken, LD50 are being used for, upon that, the doses for human are being estimated. So we have taken the blood from Fanconi anemia patients, these children, and then we made a comparative analysis with the data obtained from the parents. And the similar color reflects that they have the origin coming from the same family. And when we have tested in cyclophosphamide, we have used HepG2 macrosomal fraction. The first take-home message is that the frequency of the macronuclear increased significantly in the parents, the lower lines, as you can see. And when we look to the study, the effect in the patients, the effect was tremendously significantly higher. So this indicates that this type of drug not only may cause toxic effect in the normal cells, but the harm is going to be significantly higher in the patient. And this is the issue should be considered when the drug is being administered to the patient with known deficiency in DNA repair process. And again, this type of setup, the experiment might be very useful to do it in advance before any clinical trial to be done on patients, normal patient, as well as the patient with deficiency in DNA repair process. And of course, as I already started my talk, the human HepG2 is a very useful assay because it contains series of the phase 1 and phase 2 enzyme, but of course, it also has in some cases, the enzyme of interest, both phase 1 and phase 2 may not be present. Upon that, one of the great advantages of having hepatocytes is that different cell lines can be initiated, as has been done in the last decade, and different classes of the human hepatocytes has been generated, as illustrated here, and the comparative analysis has been done using the same type of the agent, and we can address the effect at different doses. And the interesting would be that to combine, not only generate a battery of the system, also generate a battery of the cell lines. This is one approach to generating different types of the human hepatocytes. And another interesting approach and very promising one is the application of the 3D that seems to be mimic better situation of the normal human hepatocyte. That will be the following talk by Professor Jenkins. And the last issue is that I would like to address the point that our mission of the GHI, in particular a section of the genetic toxicology and genomics, is that to expand the application of the existing method to perform the accurately human risk assessment for the chemicals we are exposed to in air, in water, in soil, and in food. And I approach you, and I need your help in order to be able to help you to do the human risk assessment better, as such to be able to improve human health. And in addition, we try to make an alternative to use of animal in genetic toxicology testing that could be beneficial from all aspects, especially the better approach and accurate approach in order to make human risk assessment. And I thank you all for your attention.
Thank you very much, Feroz. This was really insightful and interesting. I have a question for you. We were talking about human health and this model that you develop and show improved sensitivity for detecting genotoxic compounds and all of this. So in the case of liver cancer, which is the third leading cause of cancer death worldwide, what is the application of this research that we are doing for finding this, detecting liver cancer earlier maybe using molecular mechanisms, so all of these hepatoma cells for discover things, and how we can stop liver cancer earlier, detect it earlier, and stop it earlier.
It's an interesting question, but of course it's not much related to toxicology, but what it can be done, actually, it's also due to the short of time, I did not approach that one. This is actually can be done by looking for gene expression profile. The look into the gene expression profile, when we do in the human hepatoma cells, including HepG2, we get a panel of suppression or induction of the specific gene that they might be responsible for the people of getting human liver cancers. So this could be one of the interesting approach to do. Other things that we can do, though, now we are here talking using the HepG2 cells in the genotoxicology, but other issues that we have also done is that we look, when we are trying to, we can also using the same cell lines, we can improve the chemotherapy regimen, and as such that we can select the chemical of choice for treating the cancer patient having the human liver cancer, and we can approach that one. First of all, we can see whether they are sensitive to the drug of choice. This means that the normal lymphocyte from this patient can be taken, and the same type of experiment as I showed you in the previous slide can be done. We check the normal sensitivity of the normal cells, and also we can check the sensitivity of the tumor cells to the target of interest. And combination of these will be very much helpful. The first one, the gene expression profile, try to look for involvement of the genes, different genes involved. And the second will help to do the better therapy regimen.
Thank you very much. The question is, how many years will it take to use these in vitro models as an alternative to animal for assessing food safety?
We hope that to be able to do it in the 5 years, but it's going to be really, that's the point. GHI is an extremely useful platform to do that, because we need more to expand this type of information worldwide. So that's the whole issue. And via the GHI, we hope that the people can approach us in order to be able to implement the technology in the place they are doing it. Therefore, more hands are getting involved. And that would be very useful, because we do have a technology. We only need more people to do the test.
Hopefully, we'll be able to do it in 5 years' time.
Yeah, let us hope.
Thank you. Thank you very much, Ferouz. And let's go to our second speaker. He's a professor, Dr. Garrett Jenkins. He's a professor of molecular cancerogenesis at Swansea University's Medical School and an expert in DNA mutation research. He's also the Associate Dean of Research for the Faculty of Health and Life Science in Swansea University. Dr. Jenkins has degree in biophysics from King's College London, biotechnology from the University of West of England, and a PhD in molecular mutagenesis from University of Wales. His research group at Swansea investigates the mechanisms underlying DNA mutagenesis and carcinogenesis. We will hear now Professor Jenkins talks on 3D liver tissue models for DNA damage assessment.
Okay, thank you. So I'm going to talk about 3D liver models, and particularly HepG2 liver models, which follows on from Ferouz's talk. Talk about how hepatic cell lines can be used in 3D context to study genetic toxicology, and I'm going to answer the question, why do that? What is the advantage? And so 3D models are becoming more common in medicine and biology. They're revolutionizing various aspects of this. So the European Association of Cancer Research organized Goodbye Flat Biology conferences. There was one held before COVID, and there's been one held virtually since. And this phrase comes from a Nature editorial back in 2003, which described how 3D tissue models were going to revolutionize the sort of medical and biological field. And the quote at the bottom is not my quote. This is a quote from the Nature editorial. And certainly we're seeing more and more 3D models being used. So the question is why? Why use liver models? And the reason is that 2D monocultures, where you grow cells in the bottom of a tissue culture flask, aren't really representative of human physiology. And so if we want to better predict human risk, so toxicological risk, then we need better models. And it is thought that the 3D cultures of human cells will have the desired applications to represent human physiology better, because they have the cell-cell contacts, they have the cell ECM contacts, they're species specific, so we use human cells to create 3D mini tissues, 3D models, instead of using animal models, which are perhaps not equivalent to human biology. And so these 3D models in general are thought to have better representative gene expression profiles than equivalent 2D cell models, partly because they have that complex architecture, they have a gradient of nutrients, they have a gradient of O2, they have a gradient of paracrine factors and other things that are produced within them, reminiscent of what happens in vivo. So there are commercially available tools to study 3D models. Some of these are shown on the right here. It's obviously a sign of the maturity of the field when there are commercially available tools that can be used. The attraction of the commercial sector suggests that this is accepted as being a growing area of research interest. But it's important to say that the complexity of the 3D models does come with a price, not just a cost in terms of funding, but a cost in terms of time as well. And I'll touch a little bit on that in later slides. So there are many different ways of making 3D models, whether they be liver, lung, GI tract, or anything else. So the first question that's always asked is whether they're going to use primary cells and produce organoid type models, or whether they're going to be made with cell lines, which are perhaps more easily available and more cost effective. But constructing 3D models can be done in dozens and dozens of different ways. And the image here is just of some papers I was reading whilst putting this presentation together about the different approaches that can be used, the different tools that can be helpful in developing 3D models. So the reason for doing the research into 3D tissue models for genetic toxicology is to try to encourage their uptake as regulatory testing tools. So in terms of regulatory genotoxicity testing, many of you will be aware that there are two main stages. So stage 1 is carried out in vitro, and then stage 2 is usually carried out in vivo. And this is for equivocal or positive results in vitro, and that is used to confirm or to de-risk in vitro data. And the idea of the exploration of 3D tissue models is to introduce a stage 1.5 instead, which is shown on this slide here, which would use 3D human tissue models as replacements for the animal test to be able to de-risk any in vitro positive data, which was perhaps a misleading positive event. And then a much smaller number of these, which remained positive with 3D human tissues, would then go through to animal tests. So it's about amending the regulatory testing framework. So I did a little bit of research last week looking at how common the papers are being, how many papers are being published in this sphere of 3D models in the genotoxicity space. And so by putting the words 3D and genotoxic into a publication search, you can see that in recent years there's starting to be dozens of papers per year published, whereas back in 2010 there were only 2 or 3 papers being published. It's becoming much more attractive for people to look at this field. People are much more interested. And the reason they're interested is that the 3D models, the assessment of genotoxicity in them is thought to better resemble the human situation, the responses to genotoxic insults. And so therefore, from a safety assessment point of view, it may be that we could reduce some of the inherent false positive or misleading positive results that we get with traditional in vitro cell-based tests. And if we could reduce those false positives, then we reduce the animal usage, and there's a 3 R's benefit. Although I would say that this is a long journey, and we've only just started to begin it. So there's quite a while to go before the replacement is the replacement of animals with 3D models is actually realized. So the closest that we're going to get to that is with the 3D skin models, because Europe took the decision 10, 12 years ago to ban animal testing for cosmetic ingredients. So cosmetic ingredients now have to be used with the use of animal models. And this stimulated a lot of interest in producing 3D human tissue models, which could replace those animal tests. And this has been fantastic and has produced some OECD guidelines already. And the genotoxicity OECD guidelines using 3D skin models are well under construction, and this includes both the micronucleus endpoint and the COMET assay endpoint that Farouz mentioned in his talk. So there's going to be an OECD guideline for 3D skin models for both micronucleus and COMET in the coming years. So that's a fantastic step forward for 3D models in general. So what we've been doing in our research group at Swansea University is trying to understand how we might do the same thing for liver 3D models, because this is a real urgent need, and mainly because most in vitro tests suffer with poor levels of metabolism. As Farouz said, you can supplement this with S9, which comes from rodent liver, stressed rodent liver, but it's not really representative of human liver metabolism. And also S9 is not really a 3 Rs benefit, because the S9 is taken from rat liver, so it uses animals to generate the S9. And so in vitro tests don't always have fantastic metabolism, and so the idea that the 3D liver models might be even more metabolically competent than the flat 2D liver cell lines, such as Farouz was talking about earlier, means that we might get a more metabolically proficient model, which could then be much more sensitive to detect genotoxic agents in an in vitro setting. So we've developed simple hanging drop models. So we've tried to simplify the way to develop these liver spheroids, and this was deliberate because we wanted to maximize the chance that there was a transferability of this to other groups, so other groups would be able to use and try this approach. This is the key to uptake and the key to regulatory change in producing something which is widely available to other people to try out and generate data. So we've used a simple hanging drop model, which is relatively low cost, it's relatively easy to construct, and it's relatively time efficient compared to alternatives. We use a micronucleus endpoint because it is an OECD compliant endpoint, both in vitro and in vivo. Other endpoints are used by other groups looking at this sort of area. And there was a choice of cell line when we started this, and so we've tried a few different cell lines, and I'll explain to you why we settled on HepG2 in the coming slides. So there are many studies out there which have been looking at 3D liver models for a genotoxicity endpoint. Several of these have looked at the COMET assay as an endpoint with 3D liver models. We've been looking at the micronucleus endpoint, and one or two other groups have followed suit. There has been some interest in H2X gamma as a double strand break marker in in a liver spheroid model. Most of the 3D models which have been described so far for genotoxicity testing have used HepG2 cells or a derivative cell line of HepG2 cells, but some of them, particularly the COMET studies, have tended to use HepRG as well. And so there's a mixture of different cell lines available. And the interesting thing from reading these papers, this is just a representative selection of the papers that have been published so far. The interesting thing about reading them is that there's been a lot of conformity around how you put these liver spheroid models together in terms of the cell density, the length of time it takes for the spheroid to form, the sort of measures you do to ensure that the spheroid containing the cells in there are viable, and the other aspects of the technical details of how you put them together have sort of coalesced around some similar principles. So we started off asking the question, should we use HepG2 or HepRG spheroids? HepRG cells are thought to be better liver models, to be more representative of human liver than HepG2 cells, which some people claim are perhaps lacking in some of the liver metabolic enzyme expression. So we were interested in trying both those cell models out, but the HepRG models suffer because they are a differentiated cell model and they don't proliferate even when they're in a spheroidal form. And that is a real problem for the micronucleus endpoint, because those of you who know about micronucleus assay will know that you need cells to divide. It's a post-mitotic assessment of chromosome damage or chromosome copy number change. And so that meant that the HepRG cells were pretty much not feasible for this assay. And so we decided to focus on HepG2 cells. The HepG2 cells are grown they're cultured in hanging drops, inverted, and so gravity pushes the cells together into a spheroid. And you can vary the amount of cells that you put into the drop. So a 20 microliter drop might contain thousands of HepG2 cells, and you can vary that. And over a period of 4 to 7 days, they grow to being about 0.5 millimeter in size. But what's interesting is the more cells that you put in, you can create bigger spheroids. But those spheroids sometimes have a necrotic core, and you can see that in the images on the screen, hopefully, that at day 7 with 10,000 cells per 20 microliter drop, you see a black necrotic core. So lower cell number allows you to keep the cells in the spheroid viable for longer, which is important. Obviously, there was an interest in why, what are the benefits of growing HepG2 cells in a spheroidal form rather than a flat 2D culture. And it had been suggested that they become more liver-like in this manner, as do lots of other cells that are used in 3D microtissue formats. And we were certainly able to see that looking at aspartate transaminase, urea, and albumin, that compared to the same cell number of HepG2 cells grown in 2D, in a 3D structure, 3D context, these HepG2 spheroids were expressing these liver markers to a much higher degree. So they were more liver-like in that regard. But obviously, from a genetic toxicology point of view, we're really interested in metabolism of pro-mutagens, pro-carcinogens into genotoxic agents. And so cytochrome P450s as examples of metabolic enzymes are really important. And so we were also able to show that as you culture the spheroids, the HepG2 spheroids, they start expressing more and more of these cytochrome P450s. And the gel on the left shows CYP1A2, mRNA levels and N2, mRNA levels and endpoint PCR from RT-PCR. And on the right is a graph showing CYP1A1 and 1A2 comparing 2D expression levels in black to the 3D expression levels at day 4 and day 7. And huge increases in the expression of the cytochrome P450s here. So that's great. You get mRNA expression, really important. But what about activity? So we were able to look at how these both 2D and 3D HepG2 constructs were able to actively produce cytochrome P450 enzymes, which through a Omega bioassay, we could assess how active they were in metabolizing the test compounds. And not only is the mRNA expression level increased, but that translates to increased cytochrome P450 activity as well, which was really pleasing and clearly explains why there is a huge interest in growing spheroids, 3D models in general, but certainly for a liver model situation. And so we wanted to use a micronucleus assay, as I said. So we grow HepG2 cells, put them into hanging drops, turn them upside down. That forms the spheroids over a 3 to 4 day period. And then they're treated with test agent for 24 hours. And then groups of spheroids are then disaggregated using trypsin and back to single cell preparations, which are then put onto microscope slides and scored using standard scoring criteria. Of course, you can adopt this, and so we've moved to a 96-well plate format where the hanging drops, instead of being in Petri dishes, are actually on the lids of the 96-well plates. They produce lots of spheroids, which are then treated, and then we use our automated image analysis system for micronucleus detection, which is the Metaphor system from Zeiss. So it's a recent adaptation of making this a little bit more high throughput. Well, perhaps high throughput's the wrong word, increasing the throughput anyway. So lots of publications suggest that for growing 3D models in general, that hydrogels or support structures are really useful. So some people use agarose as a scaffold or a hydrogel to support its development and to maintain its spheroidal structure. Because if you put the spheroid back into the bottom of even a microplate, it will start to spread out and form a monolayer again. So the agarose can prevent that. And so this shows that the use of agarose can increase the cell viability of these models, and it also keeps them more spheroidal over time. However, there is a detriment to using things like agarose, and that is that the agarose sequesters some of the chemical that you're testing the genotoxicity of. So this is benzopyrene genotoxicity with and without agarose as a hydrogel. And you can see that without the agarose, you get a much more potent induction of DNA damage because some of that benzopyrin is getting trapped in the agarose layer at the bottom of the wells. So one of the first studies that we carried out was using benzopyrin and FIP in 2D HepG2 cells compared to liver spheroids. And if I draw your attention to the bottom graph here, so this is FIP, and this shows clearly that the 3D version, so the 3D spheroids, are much more sensitive to detecting FIP's genotoxicity in terms of micronucleus induction than the equivalent 2D HepG2 model. And so you get a positive response with FIP at 5 micromolar with the HepG2 spheroids, whereas it takes double that amount of FIP, 10 micromolar, to get a positive result with the HepG2 2D cultures. And this is probably related to the fact that the HepG2 spheroids pump out a lot more cytochrome P451A1, which is particularly important in FIP metabolism. What was unexpected was we started looking at other chemicals and their responses in 2D versus 3D, and MMS, methane sulfonate, a direct-acting alkylating agent, also shows that it is more potent in a 3D liver and spheroid model compared to a 2D HepG2 model. And there is no, that was a surprise to us because there's no metabolic involvement of cytochrome P450s in MMS activation, and so that was an unexpected observation. But it has been repeated in several other studies, both in a COMET system and in another micronucleus system. So we weren't expecting that. We've used these models to look at nanotoxicology. So my friend and colleague, Professor Shireen Dope, who's shown in the middle of this picture at the bottom right, led a European Horizon 2020 project called Patrols, developing advanced in vitro tools for nanotoxicology. And one of those tools that was developed or tested over the last 3 or 4 years has been a HepG2 spheroid system specifically for nanoparticle genotoxicity assessment. And as part of that, again, the Patrols group that were funded partly here in Swansea University looked at the activation of different chemicals and the genotoxicity of these chemicals in HepG2 2D versus 3D. We saw the same result with MMS, but we also saw that with aflatoxin, which does require metabolic activation, we saw greater genotoxicity in the 3D versus the 2D system. But that was the whole Patrols project was designed at looking at nanoparticle genotoxicity, and the data from the nanoparticle studies showed that not only were the HepG2 models really, really valuable for detecting genotoxicity, and the reason why liver models were important for nanotoxicology is that in vivo nanoparticles tend to accumulate in the liver after both inhalation and ingestion. And so what was interesting was we could use these HepG2 liver spheroid models to detect genotoxicity induced by nanoparticles. We could also do longer term dosing. So instead of 24-hour dosing, we could do up to 5-day dosing. And so these graphs show zinc oxide genotoxicity on the left from a short-term 24-hour treatment with binucleate micronuclei, and then on the right, a longer-term experiment using the mononucleate form of micronucleus assay. And for several of the nanoparticles that were tested with the HepG2 spheroids, as with some of the chemicals and as with MMS, the 3D spheroids were much more sensitive to genotoxicity compared to the 2D system. And that made us wonder about what is the cause of that. And so for nanoparticles, most of the genotoxicity is induced by reactive oxygen species and oxidative stress. The particles release ions within the cells, and that causes Fenton-type chemistry, which causes reactive oxygen species induction. And so we wondered if the spheroids themselves were perhaps on the brink of undergoing some oxidative stress, and so the nanoparticle treatments were pushing them over more easily into that oxidative stress environment where they were able to show oxidative DNA damage more so than the 2D system, which we wondered had less oxidative stress. And so we started playing around with acute and chronic treatments in 2D and 3D to pro-oxidants. And the top graph shows hydrogen peroxide, the bottom graph shows potassium bromate, 2 classic pro-oxidants. And that the right-hand graphs for both chemicals show that the chronic exposures in the 3D context give a much more potent genotoxic response compared to the acute exposures. And this wasn't seen with the HepG2 2D cultures. And so this supported the idea that there was some extra oxidative DNA damage being induced in these spheroids, and this probably is something that needs further research. And this is something that we're certainly looking at in terms of using lots of confocal approaches to look at oxidative stress, and also looking at antioxidant supplementation, which we have shown that the antioxidant supplementation increases viability of both the 2D and the 3D HepG2 cells, and reduces some of the background micronucleus, particularly in a 3D context. So this is something that's we're exploring further. One specific example that I mentioned in the abstract that's related to this event was we were interested in understanding if the HepG2 spheroid approach could tell us something about urethane. So urethane is an interesting compound. It's unique in some ways because it's a misleading negative genotoxin and carcinogen, so that in vitro, it is pretty much negative in all the genotoxicity tests that have been carried out. But it is an in vivo genotoxin and carcinogen. And so the suggestion was that the reason it was negative in vitro was that there wasn't adequate activation, metabolic activation of urethane into a genotoxic metabolite. And we certainly have studied this for a number of years in lots of different cell models and found negative results in vitro. But as you can see in the graph on this slide, that using a 3D spheroid system, a HepG2 spheroid system, we were able to show that there was a dose-dependent increase in micronucleus induction in DNA damage with urethane, albeit that that these were relatively high doses of 10 millimolar or more. So one of the things I said at the beginning was we had a specific strategy about producing a liver spheroid model that was easy to transfer and for other people to try out. And so we think this is the most important feed change regulatory practice. So part of the Patrols project was, there was a task in there which was about transferability of all of the models that were developed as part of the Patrols project. And the HepG2 liver spheroids were one of those models. And so in this, so the paper was published earlier this year. In this task, there were two naive labs that received SOPs of how to create and then how to test simple measures of cytotoxicity, viability, and some cytokine release. And the result of that transferability was that the two naive labs found it relatively easy to create the HepG2 spheroids consistently, and they were able to get viability levels and responses to toxic effects in those models to be very consistent. So the three labs, our lab as the experienced lab and the two naive labs, showed very good concordance in terms of the outputs. And several of those labs that took part in this study were able to assess genotoxicity in form of micronucleus induction, particularly for benzopyrene induced micronuclei, and they were pleasingly able to identify that the benzopyrene gave strong micronucleus induction in these systems as well. So that was a really pleasing aspect of the transferability of these models, which augurs well for them being perhaps widely used and maybe used by more people going forwards. And as part of that, postdocs working on the Patrols project wrote a paper, although that probably isn't that's probably not the way to describe it, because if any of you are familiar with the Journal of Visualized Experiments, then this is less about the paper and more about the film clip that is produced as part of this. It's an online journal that has the details of the methodology that's been described, but it is mainly based around the film clip that's produced by the team of people who come to visit your lab to generate this film clip, which shows in graphic detail how you develop the models, how you handle them, how you use them, and then how you measure an endpoint like the micronucleus assay for genotoxicity. So this was a real step forward in terms of transferability as well. And I looked at the sites on Sunday and saw that there was over 3,000 views of this particular video which is associated with the Jove paper. So that, again, is really good. That's really pleasing to see that people are taking interest in this, and hopefully it leads to more people trying these models out. So last couple of slides. So the challenges and the problems. So it's important to bear in mind the negatives as well as the positives. And so 3D models in general and HepG2 spheroids are captured within this. The throughput of these models will always be relatively low. These aren't going to be a high throughput alternative to standard cell-based in vitro testing or studies. They're always going to take longer, and the throughput is going to be lower. We have to accept that. And there is some uncertainty about which is the best and most appropriate models to study. So this is true for all 3D systems, whether they're looking at which organ or tissue they're looking at. But for the hepatology models, for the liver models, there is still some discussion about which cell line should be the most appropriate that's used. We have always suggested that the HepG2 cell line is the most suitable, but there is further validation that needs to be done on each of the endpoints that are used. So there's an issue with the disaggregation of these models. So we spend 4 days culturing HepG2 cells in the hanging drop to get them to coalesce into a spheroid. And so we spend a huge effort to try and create these spheroids and maintain them in a viable manner, because we want to get that metabolic boost that they deliver from growing in that spheroidal shape. But after we have carried out the treatment, we disaggregate that model back down to single cells, which is less than 100% efficient, takes some time, and is a little bit it's counterproductive. We've created the spheroid, and then we have to break it back down again to actually measure, in this case, micronuclei, or if you're measuring the comet tail lengths. So that is an issue that we have to deal with. So there are materials and things that you can use to create your spheroids. There are aids to creating 3D tissue models and to make them more viable. But some of those things have a cost associated with them, and some of the whatever you add into that 3D construction, it has the potential to interfere in your treatment and interfere in the action of a genotoxic chemical. The binucleate frequencies of HepG2 spheroids are half of what they are of the HepG2 cell line grown in 2D. That means you have to score twice as many cells in 3D compared to 2D. So again, this comes back to the cost in time terms of 3D compared to 2D. And we've mainly used a single cell model, so HepG2 cells grown as a monoculture, but there have been attempts. We have started looking at the incorporation of Kupffer cells or immune cells into that HepG2 spheroid to better represent the liver itself. And so there are multicellular v single cell type questions. And we and others have been really pleased when we've seen that the spheroids pump out lots of cytochrome P450s. That's great for activation of pro-carcinogens, pro-mutagens, but metabolism isn't just phase 1, it's phase 2 as well. And many of the studies haven't really factored phase 2 into the question. And whether these models are more representative of human liver is something that probably needs to be further explored. So my last slide, the 3D models in general are becoming, they're revolutionizing medicine and biology. They're going to be here to stay. And so embracing them is a really good thing. We've used a relatively simple, low-cost, single-cell spheroid using HepG2 cells, specifically because we want to encourage others to use this, so that if this can be adopted as part of a genotoxicity battery, then we think that's the right thing to do. We and others have shown that when you grow these cells in 3D, you get enhanced metabolic activity, particularly for cytochrome P450 expression and activity. And our data and data of others suggest that these spheroids using cells like HepG2 are really sensitive for genotoxicity, much more sensitive than 2D HepG2 cells. And Farouz talked about how good HepG2 cells are in 2D. I would argue that they're even better in 3D and will be a more sensitive test platform for assessing genotoxicity. But this disaggregation is an issue, and so there are alternatives to disaggregating the cells from a spheroid to then do comet or micronucleus endpoints, you can just take the DNA out of the spheroid and use next generation sequencing to find mutations. Or, as has been shown in at least one publication recently, you can use biomarkers such as H2X gamma, which you can use using confocal microscopy kind of approaches in the intact spheroid to save having to do that disaggregation. And so we'll just finish with some acknowledgements. These are the people who did the work. This is the group at Swansea University, the funding here, and on the right, the three people who have done most work around the HepG2 spheroid work, Kelzoom at the top, Jill and Sam, and I will stop there.
Thank you very much, Gareth. Very fascinating research. These models actually becoming an important and a very promising screening tools. Are these complex in vitro models can be adopted for discovery of novel targets like therapy development, I don't know, some validation experiment, probably in the area of personalized medicine or something like this?
Yeah. So I think there's a huge potential. If you just do a PubMed search for 3D tissue models, there are thousands of papers, and most of those are in developing models for assessing inter-individual variations in various different genes and proteins, which then align themselves to the personalized medicine space. So there are organoids, which are primary cells taken out of patients, which are really valuable for studying personalized medicine aspects, whether that is to chemotherapeutic agents, whether it's response to all sorts of other things. So the principles of using a 3D tissue model are the same. The developments that are happening in the toxicology field benefit from the developments that are occurring in the biomarker discovery field, in the clinical field, which are using the same kinds of approaches to generate models, whether they be primary in origin or using cell lines. So the answer to your question is yes, they've got widespread uses, and the benefit for us in the genetic toxicology world is we can use all those advances to our benefit to make our models better. Yeah,
absolutely. And because you mentioned about the new next generation in vitro models, what are these next generation? What is your prediction?
I think that multicellular 3D models are probably the thing that will develop. So, the liver isn't just made up of hepatocytes. There are many other cell types in there. And so better representing the liver with a multicellular system is probably a good thing. And I think this disaggregation, that I think the power of genomics now, the power of next generation sequencing, we won't have to disaggregate liver spheroids or any other 3D tissue to study a genotoxic endpoint, because in the years to come, we will just extract DNA, send it off to an organization that does next generation sequencing, and they will give us a profile back of, yes, this is a mutagen, and here's the profile, the signature of the kinds of mutations that will be induced. That those two things are going to be really powerful moving forward.
Very promising future. Thank you very much, Gareth, for this presentation. And we'll go to our final speaker today, Professor Dr. Robert Turecki. He is a professor at Masonic Cancer Center at the Department of Medicinal Chemistry, and he holds inaugural Mason's Chair of Cancer Causation at the University of Minnesota in the United States. Dr. Turecki received his PhD in Nutrition and Food Science from Massachusetts Institute of Technology. He conducts mechanistic research on the biochemical toxicology of dietary and environmental genotoxicants. Professor Turecki will immerse us into cooked meats and cancer etiology. Over to you and welcome.
Thank you very much for the introduction. And Firuz, thank you very much for inviting me to this excellent seminar. I believe the work that we're doing here is highly complementary to the work that both you and Gareth have reported in vitro. So my laboratory is involved in a number of different toxicology projects on chemicals, and the one that I will be focusing on today is the potential involvement of cooked meat in prostate cancer risk. So traditionally, epidemiology has been an observational science, almost like a black box. There was understandings perhaps of lifestyle factors and risk, and the final outcome is disease, or in this case, cancer, in the interest of genotoxic chemicals. However, in the last 20 years, there's been a tremendous advancement in molecular epidemiology because we have much more sensitive instruments, mass spectrometers, that can identify chemical exposures, for instance, in tobacco or carcinogens such as heterocyclic aromatic amines and cooked meats. We can measure the bioavailable doses by looking at the parent compounds or the metabolites that will be present, for instance, in blood, urine. Some compounds or the metabolites will accrue in hair and fingernails. These are biomarkers of exposure in the bioavailable dose. For genotoxic carcinogens, many have to undergo metabolism to generate reactive intermediates that will covalently bind to DNA or protein. This would be an index of the biologically effective dose. Ultimately, we want to be able to correlate, for instance, chemicals that damage DNA to form adducts that can induce mutations and link these mutations to specific mutation spectra, particularly in cancer driver genes, to try to link specific exposures to cancer. So my laboratory has done over the years a number of studies, either by animal models, in vitro cell models, working with cell biologists, and ultimately developing biomarkers that we establish in these cell lines or animal models to measure metabolites, DNA adducts, protein adducts in humans to see whether there is a good extrapolation in the biochemistry that occurs in animals or cell models to human subjects. The way we do this is by mass spectrometry. The mass spectrometer, it is our eyes. It is how we see what is happening, what's going on. We are challenged by generally getting low quantities of samples, particularly biopsy tissues. Many instances we have a wide range of analyte levels, but they're very low, parts per billion to parts per trillion. We either do absolute or semi-quantification, and with mass spectrometry, we do our best to structurally identify the biomarkers. So I was involved about 6 years ago in the IARC monographs on cooked meats, both processed and red meat, and it was concluded that processed meat was classified as a group 1 carcinogen for colorectal cancer. Red meat was classified as probably carcinogenic to humans in colorectal cancer group 2A, but the working group also noted that there was a positive association with red meat consumption with pancreatic and prostate cancer, and it's the prostate cancer work that I will be discussing today. So I use this analogy, but I need to be careful about it. Most of the carcinogens that form in tobacco occur during the high temperature burning, the combustion of the tobacco. Similarly, when you cook meats, beef, poultry, fish, if you cook these meats at high temperatures, which are often done to produce the flavors and aromas, unfortunately, they can produce low amounts, but potentially cancer-causing agents such as the heterocyclic amines, including FIP, that we have heard about already today. So prostate cancer is the second leading cause of cancer-related deaths for men in the United States, and it's one of the most prevalent cancers worldwide. So our questions are, what chemicals in cooked meat cause DNA damage of the prostate genome? And does, in fact, eating a lot of red, cooked, well-done meat explain elevated prostate cancer risk? So more than 25 heterocyclic amines are produced in well-done cooked meats. Some of them are derived from the pyrolysis of amino acids, such as tryptophan or glutamic acid. Others, called iminoazoarines, of which FIP is part of this class, are part of the chemical that's formed is derived from creatinine, which is an ATP-bound muscle reserve, and it's only found principally in muscle tissue. It's not found in plants. And I will be focusing most of our work on FIP today. So again, red meat, it's classified as probably carcinogenic to humans. We know that FIP forms in well-done cooked meats. It's been classified many years ago as a Group 2B carcinogen. My opinion, based upon the data today, would probably be upgraded to 2A, except for the fact that perhaps there's a question about the total dose, whether the actual exposures are sufficient to induce cancers. FIP is the only known mutagen in cooked meat that induces prostate tumors in rodent models. We believe that FIP's carcinogenic effects are driven by its capacity to form DNA adducts that induce mutations. And one of the questions we want to know, does FIP contribute to human prostate cancer? So this is barbecue cooking 101. This is a well-done cooked sirloin steak on a charcoal grill. I wouldn't advise people to cook their meat this well done. That's going to contain the highest levels of heterocyclic amines, including FIP. The levels that would be present in meat that's cooked medium rare to medium is probably 5 to 10-fold lower. So with respect towards HAAs, that's a safer type of meat to eat. Similarly, in bacon, there's a lot of heterocyclic amines, particularly FIP, that's formed in well-done, cooked, crispy bacon. It's safer to eat bacon as depicted on the left panel of this photo. There's considerably less FIP in this type of cooked bacon. So bioactivation of FIP, the major pathway of the sole pathway of activation that we know is CYP1A2 catalyzed N-oxidation, principally in the liver, or 1A1, 1B1 in extrahepatic tissues. This can directly react with DNA or following phase 2 enzyme activation with N-acetyltransferases or sulfaltransferases generate an even more reactive intermediate that will form a DNA adduct. Some portion of the adducts may escape repair, and if so, during cell division, this is a mutagenic lesion and can indeed induce a mutation. It could also induce cell death. Reactive intermediates can also form covalent adducts with proteins. And of course, you have detoxification mechanisms as well. So this is some work that I did over 20 years ago in collaboration with Fred Gingrich. And we talked about the discrepancies between animal models and humans. And this is an excellent example. The rat CYP1A2 liver expression is very low. It's between 10 to 20 picomoles per microsomal protein. And you can see the humans, there's a wide range in expression. We believe that this is not due to differences in SNPs that are involved in inducibility, because they're very low penetrant alleles. We think it's exposures to things in the diet, tobacco, and so forth, that are actually inducing the expression. And you can see it varies by well over 50-fold. If we look at the rates of bioactivation in the lower corner here, for both methyl IQX and for FIP, you can see that the rat activity is extremely low, and we get a nice dose-dependent response as a function of enzyme expression with rates of N-oxidation for both compounds. So the rat model is very poor in comparison to humans and does not account for the large inter-individual variation in CYP1A2 expression. We've done enzyme kinetic parameters with recombinant human CYP1A2 or purified CYP1A2 provided by Fred Gingrich. This is data on methoxyresirufin, which is O-demethylated catalysis is the end product. And you can see the kinetic parameters for human and the rat are quite similar. In contrast, for N-oxidation of FIP, what you can see is that the Km is about 12, 14, 15 times lower for humans than it is for the rat enzyme, and the turnover is much higher. So FIP activity is considerably greater in human liver CYP1A2 than in the rat CYP1A2. For MEIQX, you can see the Kms are similar, but the turnover is much, much lower in the rat model. So again, these are isolated enzymes, but these types of parameters and considerations in catalytic activity must be considered in human risk assessment. So let's go on to prostate cancer etiology. Age is the most important factor. It's a cancer of the elderly, and I say elderly, and I'm already 65, so I don't actually like that term. African Americans have higher risk than do Caucasian Americans. There is inconsistent data about the role of pesticides and endocrine disruptors on PC risk. But again, what I'm going to focus on today is for the red meat. So the epidemiology data on dietary factors and PC risk, they're not consistent. And our hope is by having biomarkers of chemical exposure and DNA damage, we may understand what chemicals in the diet may be contributing to damage the prostate genome and prostate risk. So we have done several semi-controlled feeding studies where we've given well-done cooked meat with varying levels of FIP in the diet. We phenotyped for CYP1A2 using the caffeine metabolic ratio. We had graduate students eat well-done cooked meat 5 times a week for 4 weeks. And we've characterized a number of these different biomarkers from the FIP DNA addict to urinary metabolites. We've looked at a specific addict of FIP form with serum albumin. And interestingly, a small but sufficient amount of unmetabolized FIP, when it goes through systemic circulation, binds to melanin or other pigments in the newly grown hair follicle, and depicted as these little yellow balls, grows out in the cortex. So once you can snip the hair, digest the hair, and you can measure FIP. In contrast, many of the other heterocyclic amines, their binding affinity is considerably lower. I need to emphasize this is the non-metabolized dose. And we originally did work in, this is another pilot study actually before we did that controlled feeding study. And originally we did not have vegetarians, but we had dogs. And this is my pet dog, Moses. And to my surprise, we found incredibly high levels of FIP in their fur. Interestingly, almost all of it was accrued in the black fur, not the white fur, showing that pigmentation is important for binding of FIP. And then, of course, we did ultimately have vegetarians, and you can see the vegetarians have virtually no FIP in their hair, which makes sense because, again, this carcinogen is produced in well-done cooked meats, poultry, and fish. I've also wondered whether the high-temperature kibble, which these dogs ate, whether this is actually contributing to canine cancers, because these dogs eat this food every single day for their lives. So going back to the controlled feeding study that was done with the University of Hawaii, we collected blood, urine, and hair at different time points. So for the hair study, we collected during the pre-feeding stage where we asked the individuals to refrain from eating cooked meat. We clipped the hair from the nape of the neck behind the earlobe, so this was newly grown hair. And then after 4 weeks of feeding, we collected the newly grown hair again at the nape of the neck. And then we had a washout period where we asked them to refrain from eating well-done cooked meats. And what you can see here is we have a nice dose response in the accrual of FIP and hair as a function of the amount of FIP that they ate 5 days a week for 4 weeks. And we have normalized this for melanin contents. And I believe that this is a better or a superior way of assessing exposures in the food frequency questionnaires, in which levels of FIP in cooked meats can vary by over 100-fold. So this is the sole long-lived biomarker of FIP, but also other HAAs to assess dietary exposure. However, again, it's the unmetabolized compound, and it's not the biologically effective dose. So in the same feeding study, we've also looked for addicts formed of the cysteine 34 serum albumin. This is a hot spot. It binds a number of reactive electrophiles of toxicants, including FIP. And this is some of the chemistry that we've worked out. The reactive intermediate is the hydroxylamine, but this is a reducing agent in vivo, and it gets oxidized to the nitroso intermediate. And that can form a semi-mercaptile with the cysteine 34, which rearranges to the sulfonamide. And we have characterized this. We have digested out the protein in vitro with trypsin and chymotrypsin. And the chemistry of this linkage is such that if you treat isolated plasma or albumin with acid, you're going to cleave this adduct, and you're going to generate the parentamine and the cysteine sulfonic acid. And this you can fastly extract from albumin and analyze it by LC mass spectrometry. And so again, we did that controlled feeding study for 4 weeks. And again, the pre-feeding study, we collected blood from subjects on the free choice diet. We had one outlier, but otherwise the levels of this adduct were detectable, but low. And then after 2 weeks and 4 weeks of feeding, you can see the level of adduct jumps up. We had 2 different cohorts, 2 different times, and the same pattern occurred. However, after cessation at 63 days, we've lost 90% of the adduct. And it actually turns out, unfortunately for us, the adduct only has a half-life of 3 days. It's going to make it difficult for us to use this biomarker in human studies. However, the data does indeed prove that FIP gets bioactivated in humans to a reactive intermediate. And interesting, the levels of the adduct at day 48 they vary by over 200-fold, but it's not associated with CYP1A2 activity. Using the metabolic phenotype ratio of caffeine in our hands, this expression varies within individual by at least 2 to 3-fold. Again, so this is going to be somewhat challenging, and we do not have an association with activity with the level of the protein adduct. Again, its half-life is such that it's not going to be readily employed on subjects on a free choice diet. We've characterized the metabolites of PhIP in human urine. The major pathway of metabolism is through bioactivation of the N-hydroxy to form glucuronide conjugates. This is over 75% of the ingested dose, so bioactivation is very efficient. And again, the unmetabolized PhIP in hair is a long-lived biomarker. So we asked the question whether any of these biomarkers can be employed in cancer risk assessment. And we have recently completed a study here at the University of Minnesota. We had 325 prostate patients, many of which had prostate cancer. Others had bladder cancer, in which the normal benign prostate was also removed for medical reasons. And we also had subjects who had benign prostatic hyperplasia. And we looked at the biomarker of PhIP in hair. We've conducted measurements of DNA adducts in the prostate genome, as well as some oxidative stress markers, since oxidative stress is thought to be contributing to prostate cancer. So we know that we can measure PhIP in hair, healthy adults. We have data showing that some prostate cancer patients have their DNA damaged by PhIP. And we want to understand whether elevated levels in PhIP in hair may correlate to aggressive prostate cancer. Our long-term hope or goal is that if we can take a snippet of hair here, when someone is younger, we can tell them if they have higher levels of PhIP that, hey, maybe they ought to change their diet. It may reduce their risk for aggressive prostate cancer later in life. So we actually measured the levels of PhIP in hair of these 300 plus patients, and there was a self-reported administered food frequency questionnaire. And you can see some subjects have quite a bit of PhIP in their hair when normalized per melanin or simply per gram of hair, but our correlations with the self-reported intake of meats per week is poor. And actually, I ascribe that to either two reasons. One is because the levels of PhIP can vary highly in cooked meats, and each one of us has a definition of well-done cooked meat that's different. And the other, well, I believe that's the major reason for this. And also, we do not actually have the specific quantity levels that people ate per week. So that is another variable. Our method is such that we've looked at PhIP levels in hair of several subjects over 6 months, and the levels don't vary by more than 25%. So our analytical method and reproducibility are quite robust. So we looked at the correlation of PhIP in hair with, firstly, we looked at prostate cancer patients, again, bladder cancer patients, where the normally benign prostate was also removed, and prostate benign hyperplasia patients. And you can see that a considerable amount of PhIP is at very low levels. If I zoom in on the scale, what you can see is clearly the levels of PhIP in hair of the prostate cancer patients are higher than bladder cancer or prostatically benign hyperplasia patients. And this is statistically significant. Prostate-specific antigen, or PSA, is one of the biomarkers that are used for assessing prostate cancer risk. It's a controversial biomarker, and it's not 100% concordance with PC risk. Generally, however, it's still commonly used, and generally, as your PSA levels increase above 4, your risk for high-grade disease and cancer is elevated. And if we look at, we had to break this down, made it binary. So the threshold cutoff level of moderate to aggressive is viewed as 4. What you can see is patients with moderate to aggressive PSA scores have significantly higher levels of PhIP in their hair than do patients at levels below 4. The Gleason score is another factor that is used in characterizing the aggressiveness of the prostate cancer disease. And Gleason score of less than 6 is usually considered benign. Gleason scores of 7 and above, there's generally treatment, whether either surgery or radiation or hormonal therapy. And what you can see is significant increase in FIP hair levels in patients with the elevated Gleason scores. So this is the first biomarker to link well-done cooked meat with aggressive prostate cancer risk. Now, whether FIP is a causative agent is another story, but this does support some of the epidemiology that indicates aggressive PC for subjects who are eating well-done cooked meats. And this is a histogram of our cohort. You can see that it's skewed to the left, and actually our cohort is in general exposed to low levels of FIP. And remarkably, we are seeing associations. We saw the FIP addict in about 10% of the cohort, and oxidative stress markers from acrolein in 30 and 47%. And interestingly, acrolein this addict was not correlated to the prostate pathology biomarkers as was FIP in hair. And we looked for other cooked meat carcinogens. The only addict we've been able to measure thus far was DGCA FIP. With those patients which we had levels of FIP in hair, we did a regression curve to see whether there was a correlation with FIP levels in hair with the levels of DNA addicts. It's approaching significance. It was actually stronger for hair than it was for melanin. But again, our percent of the cohort, it's not that high. We need more subject numbers. This is a challenging thing to do, obviously, to get prostate tissues. Also, we don't know when the last time the subjects ate cooked meat. They could have formed an addict, and they could have been repaired before we retrieved the biospecimen for analysis. We've also done untargeted DNA addictomics. Most DNA addicts, when we fragment them, it's called tandem mass spectrometry, they lose the sugar molecule of the addict to lose 116. And so this is a targeted analysis, but we asked the question, well, can we screen for addicts by non-targeted methods where we look for compounds after the DNA is digested that lose the deoxyribose moiety? And we've done some chemometric analysis, and we looked at patients with high and low Gleason score. This is a small pilot study, N equals 7 for each. And these are volcano plots. And what I want to emphasize is in this lipid peroxidation fraction of DNA damage, this green dot actually represents a formaldehyde addict that is elevated in subjects with high Gleason score versus low Gleason score. And its variable importance trajectory shows clearly it's significantly higher in high Gleason score patients. We also identified some novel lipid peroxidation 4-hydroxy-2-alkenyl addicts, about 5 of them, and we believe they are rearranged structures from this original form. And you can see many of them are elevated in high Gleason score patients than low. And again, this supports some of the observations about oxidative stress, aggressive prostate cancer risk. We've done some work in LN-cap cells, which are a cancerous cell line. And I want to emphasize that, as has already been alluded to before, that the panel of enzymes, both phase 1 and phase 2, definitely do not represent what is actually occurring in a prostate epithelial cell. The parent compounds are not activated in this cell line or do not cause DNA damage, but the synthetic genotoxic metabolites, what we can see here is that FIP is a very potent cytotoxicant in this cell line compared to 3 other genotoxic heterocyclic amines, which were also formed in cooked meats. And again, the doses that were given were such that the levels of FIP DNA damage, DNA addicts, are much higher than these other heterocyclic amines. Again, consistent with some of the prostate data in vivo, this is the only addict that we've seen thus far. And we can see that addict formation occurs down to levels approaching human exposure. So we're quite excited about this data and a possible role for FIP in cooked meat and prostate cancer. So the working model is we think most of the FIP is bioactivated in the liver by CYP1A2, goes through the bloodstream, ultimately gets to the prostate where phase 2 enzymes make reactive, even more reactive intermediates to conform addicts with protein. Again, we'd like to better understand the detoxification mechanisms in the prostate. So I've talked about meat. I don't want people getting paranoid. There are many chemoprotective agents in the diet, and we've seen some of the data in cell studies. Cruciferous vegetables are beneficial. I like red wine, although I try to limit my amounts because ethanol is a carcinogen, but there are antioxidants and polyphenols that can either induce enzymes or act as scavengers. Interestingly, garlic cloves is protective against colorectal and prostate cancer. Perhaps the most potent chemoprotective agent or beverage or food based upon epi studies is actually coffee. So key takeaways, if you enjoy eating meat, I'm not saying that you should stop. I still enjoy meat. I just will not eat it well done or charred. Eating it is cooked at medium to medium rare is safer. The FIP inhibitor is the first biomarker to link well-done cooked meat with aggressive prostate cancer risk. Our oxidative DNA damage suggests that oxidative stress is also involved in aggressive PC. Glucuronol transferases are important enzymes in FIP detoxification. Some of our data, biomarker data in humans, has been used by regulatory agencies such as IARC in public health recommendations to the public for healthy diet. Changing the public's perception about meat and healthy diet, it's a long-term goal. Man has been eating cooked meat for over 1 million years since the advent of fire. I think they were more worried about being eaten up by a saber-toothed tiger than they were of developing prostate cancer or colorectal cancer from cooked meat. But if we can educate the public a bit more to change their dietary habits, I think that would be a very powerful chemoprotective approach. So I'd like to thank my lab for their collaborations, our oncological surgeons, Dr. Wade and Murugan, our statistician, Loic Marchand for the study at the University of Hawaii, our funding, and the Masonic Cancer Chair and their support for my research. So thank you very much for your time.
Thank you, Robert. I really like your message, changing dietary habits and actually reducing meat consumption probably, and probably reducing the charcoaling of the meat people cook. I was interested, why Afro-Americans are more prone to develop prostate cancer. You mentioned this at the beginning of your talk.
That's a good question. That's still under debate. There are certainly biological, genetic reasons that African Americans are more susceptible. Interestingly, though, there is data in the literature. It's limited, but it is thought that African Americans, men, do eat certain types of meats more frequently than do Caucasians, including grilled bacon, sausages, and preferences for higher temperature than do Caucasians. Thus, diet may be a contributing factor. The biomarkers that we have established, we hope that we can advance if we get more funding to understand some of the ethnic differences in prostate cancer disparity. I might add that anecdotal, but 2 of the patients who had the highest levels of FIP DNA addicts, they were African Americans. So there may be something there about the diet.
Yeah, this will be really good to have this kind of research in the future. You mentioned that we have to cook our meat less, not too much, but does cooking methods also could modify the magnitude of the cancerogenic risks?
There is some epidemiology. Again, I'm not an epidemiologist, right? And I get frustrated with epidemiology because you have one conclusion one week and then the next month, or you can have another study that says the opposite. But there are some studies that do link the aggressive prostate cancer risk with well-done cooked meat, but not meat that's cooked medium or medium rare. But those studies, they're limited in number, but it goes, their paradigm or our biomarker data do support their paradigm. But again, our biomarkers is relatively new, and it would be really good to get this biomarker, and we're trying to do this in other cohorts and better understand people's cooking preferences. Have the cohort we had at University of Minnesota, because of the general composition of the cohort here, was over 90% Caucasian. So we're looking at other cohorts in different geographical areas of the states where there's going to be a greater proportion of African Americans and Hispanics. And again, if we can understand what these cohorts eat by the biomarker and their associations with prostate pathology, that may give us a closer understanding about how people eat cooked meats, their diet, their ethnicities, and risk.
Yeah, and is there any difference in if they're not, baking it in oven or boiling it or anything? Are there any differences?
Well, certainly for levels of exposure. So again, these compounds, they're formed at high temperatures in which the water content, the moisture, is being driven off. So you would not find these heterocyclic amines in meat that's poached or stewed. It really needs to be on a grilled surface or barbecue. Possibly broiling in the oven could contribute some, however.
Okay, interesting. Thank you very much for this wonderful presentation. We will go to our next poll. So Gerhard, could you please display the poll? Yeah, based on existing information to improve food safety standards in future, which sector should revise the current regimen? You have a minute to answer, another 30 seconds. What is the answer?
We have now for academics 20%, governments 40%, and also 40% for all of the above.
So Robert, what do you suggest is the right answer?
That's a difficult question. I mean, from my perspective, academics needs to take the lead in understanding mechanisms, whether it's in vitro. Well, in vitro methods alone are insufficient. I think the types of cell models that are being established that we've heard about today are an important advancement and get us closer to humans, and again are superior to the animal models. So I mean, academic is critical to drive this. Ultimately, though, regulatory agencies have to understand the technologies and whether they are satisfactory for assessing safety and replacing the animal models and for being used in risk assessments. And certainly other agencies such as IARC or other agencies, the National Cancer Center here in the United States, in Japan, and other agencies worldwide are critical in this involvement. So I guess it's for me, it's really a team effort. And certainly I used to work in industries. Industry also has an important role to play, particularly, I mean, the food industry, but also agriculture industry. They're producing chemicals that they're selling to the public for either direct consumption or indirect through fertilizer and so forth. So, I mean, they need to make sure that the products are safe as well. So for me, it's all the above. But I think academics needs to be a driving force. And some of the students, graduates and postdocs, they're ultimately going into industry and government as well. So
I actually agree that it's all of the above, because academics could discover things, but really the government needs to support them, and the industry also needs to do their part in everything.
Yes.
So thank you very much. I think that our webinar is at its end. Thank you, Dr. Feroz Darudi. He's a chair of GHI Genetic Toxicology and Genomics Working Group. Thank you, Professor Dr. Garrett Jenkins from Swansea University, Institute of Life Science, Swansea in the United Kingdom. And thank you, Professor Robert Tureski from the Masonic Cancer Center and Department of Medical Chemistry at University of Minnesota, the United States, for your time spent with us and for the great contribution and knowledgeable presentations. You are all wonderful, and I trust our audience, and we learned a lot about issues related to genetic toxicology and genomics. Many answers were given, so I think that was a good contribution. Again, huge thanks to all of the people in our team that have worked hard to make our webinar to come true. Thank you, Dr. Nicholas Stanley, our GHI Communication Director, for putting all the bits and pieces for this webinar together. Dr. Gerhard Schleining, for your patience and for the invaluable technical and other assistance and helping us with our interactive poll. Thank you all for your great help. Thanks goes to our audience as well, and also to the wonderful Genetic Toxicology and Genomics Working Group. Thank you very much for being with us, and see you in the next 3 upcoming webinars till the end of the year. Have a great day and or evening or morning. Stay safe, healthy, happy, and curious.
Bye-bye.
Bye-bye.
Bye-bye.